This is the University of Otago.
Wow. Professor Seddon, you know how to pull a crowd here. My name is Harlene Hain, and I'm the Vice Chancellor here at the University of Otago, and it is my great pleasure to be here with you this evening for this inaugural professorial lecture. It is really fantastic to see so many people in the audience, and you know that we've actually reached capacity, and we have folks next door who are watching this on live stream. And I think that's really an indication about the popularity of this particular professor, as well as the community interest in his area of research. So on behalf of the university, I would like to welcome family, friends, academic and general staff, and members of the Dunedin community who have joined with us this evening to celebrate. Now, as you know, in order to be promoted to the rank of professor here at the University of Otago, an academic staff member must demonstrate excellence in teaching, in research, and in service. But against that backdrop of excellence, there are a few things that stand out, particularly for any given individual. And in terms of Professor Seddon's portfolio, there are two things that I would like to highlight for you this evening. First, he is obviously a gifted graduate student supervisor. At the time that he was promoted to this position, he had supervised 12 PhD students to completion, and he was currently supervising 7 more. He had also supervised 20 master's students to completion, and he had 3 additional master's students in progress. And not only has he successfully supervised these students in order to gain their degrees, but along the way, he has also helped them to publish the products of their research. Now, many of the senior people who are in this audience this evening, and certainly the people who are sitting down here, that commitment to graduate student research, I think, resonates with each and every one of us. Professor Seddon's passion for postgraduate supervision is something that I particularly empathize with. And for many of us in this audience this evening, it was the opportunity to supervise postgraduate students that actually drew us to this profession in the first place. And for those of us who now have different kinds of jobs, it is the one thing that we continue to jealously guard amongst the midst of our other administrative responsibilities. But I think most importantly, for the postgraduate students in this room, and I suspect that there are a large number of you, I'm sure that you will agree that there is no more valuable resource during the course of your graduate career than having a smart, energetic, and highly committed supervisor. And I'm sure that Professor Seddon's students who are here in the audience this evening will agree that they have been extraordinarily privileged to have the opportunity to work with him. Now, the second thing I want to highlight about his career is that Professor Seddon has also made a very concerted effort to translate his outstanding science into practice. And in the field of the reintroduction biology, he has published a number of key papers, book chapters, and books. But more importantly, perhaps, he has produced a framework for the reintroduction of endangered species that is fast becoming the internationally recognized best practice in the field. At Otago, we are extremely proud of the academic caliber of our professoriate. But we are also extremely proud of people like Professor Seddon, who actually make an effort to solve real-world problems, not only here in New Zealand, but around the world. So on behalf of the University of Otago, I would like to warmly congratulate Professor Seddon on his promotion to professor, and I would now like to invite Professor Keith Hunter, the Pro-Vice Chancellor of Sciences, to tell us a bit more about his path to this honor. Thank you.
Thank you, Vice-Chancellor, and let me say that this is one of the more pleasurable parts of my job, introducing new professors in their inaugural lecture. Phil Seddon was born in Newcastle upon Tyne to Kiwi parents who were on their OE. But he came back to New Zealand when he was only 3, by ship in fact, and one of his earliest memories is being terrified and hiding under a table when the ship's crew sang him happy birthday. He was educated in Auckland and Wellington and then attended Otago University from 1981. And he completed a BSc Honours in 1984 and his PhD in 1988. His PhD research was on yellow-eyed penguins, and his supervisor, Lloyd Davis, who's here, had the habit of clustering his PhD students together in one room. So Phil was thrown together with Jolanda van Heesek in a shared office. Now this predated the days of health and safety, or at least serious health and safety. So their shared office doubled as a lab, and Phil had to eat his lunch while Jolanda sorted out penguin regurgitation samples. I hope you didn't get confused. At the end of the PhDs, they both set off to, in 1989, for 3 years of postdoc work at the University of Cape Town in South Africa. And the South African work also involved penguins, but it was distinguished by being the time when South Africa was undergoing huge social change. And so Phil and Jolanda were in Cape Town when Nelson Mandela was released after 26 years of imprisonment. And they joined a huge crowd in Cape Town to listen to Mandela's first speech in 3 decades. And at one point, a crazy man with a knife started slashing at the people around them. And Phil, being taller than Jolanda, could see this knife wielder coming. So this was his hero moment, which had arrived at last. And he seized Jolanda and attempted to pull her backwards to safety. The problem was that he was wearing jandals. And so he slipped over, and to this day, Jolanda is known as Phil's bodyguard. Despite this misunderstanding, a job offer from the Middle East came up for Phil, and that necessitated a quickie wedding, which was held in the Wynberg District Court in South Africa, with, as he describes, 3 guests and wedding photos surrounded by posters entitled, "Know Your Terrorist Weapons." So during 9 years in Saudi Arabia, Phil and Jolanda worked together in the vast desert-protected areas there and were involved in restoration programs for species such as the Arabian oryx. So in 2001, with a young son added to the family, Phil and Jolanda returned to New Zealand to take up a shared position at the University of Otago in the Department of Zoology. And at the same time, he took up the directorship of the Wildlife Management Program. So Phil, congratulations from me on your richly deserved promotion to Professor, and I now invite you to present your inaugural lecture.
Thank you, Keith, Vice-Chancellor, and thank you everyone for coming and sharing this time with me. A very close friend of mine who was not part of the university was congratulating me on this promotion. They said, "Oh, now you're a professor. What's the difference? Does it mean you don't have to do any teaching?" And I said, "Well, no, we do teaching." He said, "Well, does the university give you a big research grant?" And I said, "Well, no, it doesn't happen either." And he said, "Well, what?" And I had to think about it, and the only difference I could tell was that I was on 3 new committees. One of those committees was in commerce, and I went across there. I was really keen to kind of interact with colleagues in other disciplines. Went in, introduced myself to the chair of the meeting, and he turned around and he said, accused me of being an animal racist. And it turns out this was because I privileged things like native species like kiwi and kakapo over introduced species like possums. My immediate response was to threaten to go out and set some possum traps. But in the wider sense, I think it says something interesting. I think we are losing sight generally about what's native, what's not, what's supposed to be here, what shouldn't be here, what's an invasive species. We're starting to become alienated from the natural world that kind of sits around us. And it's not surprising, really, when you consider that fairly recently, global population passed a milestone. So that red line is the proportion of the world's population that's in urban areas. So there are more people live in cities than in rural areas. And the projection is this is only going to get more and more intensified. So the darker the colour here, you can see the more urbanised that area. What this does is it moves us into a very artificial environment. We're away from the natural world. We lose touch with it. We become alienated from it in a number of ways. It's not helped by the fact that we're losing natural biodiversity. We're losing species all the time. This rather compelling graphic just recently came out. So the lighter the color, the more red, the more species at risk of extinction in terms of looking at mammals, amphibians, and bird species. So despite decades of traditional conservation action, we're not winning that battle. What I mean by traditional action, it's like creating protected areas and trying to preserve species where they are. We're losing that battle. And we're moving into an era where we need to consider much more intensive kinds of conservation tools. And that's what I want to talk to you tonight about. So one of those tools out there is conservation translocation. So a translocation is really where you move species from one area and you release them in another. So a conservation translocation is where you do this for some kind of conservation benefit. And a subset of that I'll be talking about is reintroductions. So a reintroduction is where you've lost species from one area of their range and you want to translocate them back in there and try and re-establish a population, a viable population. I also want to embed this in the idea that these kinds of conservation actions are a way to re-engage people with their natural heritage. We can think about New Zealand. New Zealand has the dubious distinction of being an extinction capital. We've lost a lot of species. And if you talk to people, people say, yes, well, of course, we lost moa and we lost things like that in two waves of kind of human colonization. But it's probably not realized just how urgent, how recent, how ongoing things are. I think in most people's mind, we feel that we've got over those extinctions. We've now got Department of Conservation. They're doing lots of good stuff. They're dropping a lot of poison out of the air in order to deal with pests. So pose a question. What was the date of extinction of kakapo on mainland New Zealand? So kakapo used to be distributed widely across North and South Islands. We now only have relic population, a really small remnant population on a few offshore islands that don't have predators. Was it 1875? So this is well after kind of organized colonization. We had stoats, we had rats, we had cats, we had all these sorts of things. No, it wasn't, but we lost the New Zealand quail about then. What about turn of the century, around 1900? Not yet, but we'd lost the Stevens Island wren at that time. What about just before World War I, or around World War I? So getting into, nearer history. Not then, but we lost laughing owl around that time. Let's pull it right up close. 1965, that's certainly within my lifetime. Surely, we would have had any losses we were going to get, we'd get by then. No, but we lost the greater short-tailed bat, and this is the only known photo, the only photo that was ever taken. It was lost so fast. So when was it? 1987, we lost the last kakapo from mainland in Fiordland. And this is the last kakapo from Fiordland. So it's been held by Don Merton, who sadly passed away in 2011. Don was a pioneer of using conservation translocations, reintroductions, responsible for saving probably 3 species of New Zealand bird: kakapo, Chatham Island black robin, and saddleback. I actually first met Don when we were both in Cincinnati Zoo looking at a rather forlorn display of a stuffed pigeon. This is Martha, this last passenger pigeon. Martha died almost exactly 100 years ago today. Martha's going to play a part in the story tonight. We'll come back to Martha. The other thing I note there is this idea of you always name the last few remaining species that you have around. So if you're a species, an animal like that, and someone names you, then you better watch out. And this last Fiordland kakapo was named. It was named Richard Henry kakapo. Richard Henry, the kakapo on the end, to distinguish him from his namesake, which was Richard Henry. So this was an Irish immigrant, worked in Australia and New Zealand. A keen, self-taught naturalist, was made a warden of Resolution Island down in Fiordland. And he was appalled to see the arrival of stoats invading from their release sites in the east, coming into the west area. And where the stoats came, they started to wipe out the local birds. And he took it upon himself to try and save as many of these as he could by catching vast numbers, more than 700 kiwi and kakapo, which tells you something about the densities they were at, and moving them from the mainland to offshore Resolution Island. So a conservation translocation. Unfortunately, stoats invaded Resolution. It was too close to the mainland. They could swim that. And all his work was for nothing. All those birds died in the end. And heartbroken, Richard Henry left Resolution, never returned there, did some other jobs around Kapiti, but eventually died in an insane asylum in Auckland. Now, for a long time, I've proudly used Richard Henry as saying this is the first conservation translocation. It was in New Zealand. We're pioneers with this, until someone pointed out that around about the same time that Richard Henry was swatting sandflies in Fiordland, a man called Edward McElhenney, who's probably best known for the ubiquitous Tabasco sauce, he created a wildlife refuge, a bird refuge called Bird City in Louisiana, and took it upon himself to try and rescue from extinction the snowy egret that was being threatened by trade for feathers for hats. And he started a captive breeding program and was releasing them back. So he was reintroducing these birds back into that area and probably single-handedly saved the bird. So these were kind of individual efforts, individual people taking it upon themselves to do something. The first institutional attempt to reintroduce species was with American bison. So 1905, there was a society formed, the American Bison Society, at Bronx Zoo. The image there, the black and white image there, is a pile of bison skulls. So bison used to occupy the Central Plains in vast numbers. 70 million was the rough estimate. And they were wiped out to the point that there were fewer than 1,000, 500 and something animals left by the time 1905. This was a fairly systematic hunting. People were using the meat, the skins, they ground up the bones for fertilizer, but that was almost incidental. It was actually an engineered harvest, if you like, to put pressure on Plains Indians who were within that area to open up the area for cattle. So up to 2.5 million bison a year were being killed at one period. So the American Bison Society actually undertook the first institutional reintroduction, if you like, when in 1907 they transported 15 animals from Bronx Zoo to a release site, and that started the restoration of these species. We need to jump forward a few decades before institutional reintroductions really took off. So think about the 60s, 70s, and 80s. So golden lion tamarin in Brazil, the peregrine falcon in North America, and the Arabian oryx in Oman. That last one being headed by Mark Stanley Price, who became the chair of the World Conservation Union's Reintroduction Specialist Group and wrote a very influential book. There's been a huge number of species that have been translocated for conservation purposes, have been reintroduced. This is the tiniest kind of selection. So a lot of activity, a lot of information out there. We can summarize this a bit with this figure, which has really got two parts. So if we look at the graph at the top there, those dark bars are species that have been translocated, and the pale bars are the proportions of species in nature. So what this is telling us is that there's a taxonomic bias in what we choose to work on. And there's some other work I did with some colleagues looking at the fact that we tend to focus on birds and mammals to the exclusion of other things, almost irrespective of their threat status. I mean, there's no mystery why. We do it for conservation research, we do it for general research. There's a focus towards these things, and it's because they're cute. You can get public support, you can get money, you can do things like that. Second part of this figure is these arrows, these circles down here, which are just telling us the proportions. So there's a little over a quarter of translocations that have taken place in Australia and New Zealand. So we really are a kind of a reintroduction hotspot, if you like. There was a recent study done that said 55 New Zealand bird species, just thinking about birds, have been translocated, over 1,000 releases. This is over 60% of our entire avifauna has actually been translocated. And there are some 5 species that only exist with us now. We can only enjoy them now because they've been rescued using these kind of translocations. A lot of those early efforts, right throughout the world, were very much management exercises. And they were done without necessarily a lot of planning, a lot of thought, a lot of follow-up actually to actually find out what was going on. And with a couple of colleagues, so Doug Armstrong from Massey, Richard Maloney, who's Department of Conservation, the three of us kind of bridged the gap between research and management. And in a couple of papers, we were trying to set out a way for what we call developing the science behind reintroductions, or really the discipline of reintroduction biology, talking about better use of experiments rather than just pure management exercises, use of modelling, predictive modelling, and thinking about strategic directions, actually answering specific questions. A lot of this thinking came together in a book a couple of years ago. Excuse the grainy conference photo on the side there. But these are the editors. We're all either Kiwis or New Zealand based. So Doug Armstrong, John Ewan, me, and Kevin Parker. I've been talking a lot about reintroduction. And the thing about reintroduction is that it's a restoration. And then the question is saying, well, restore to what? What is it? What's your target state? How far back do you go? Do you go 10,000 years back? Well, a lot of these species just aren't with us anymore. What about 1,000 years ago? What about the changes since then? Or do you go 100 years ago? It's really an arbitrary decision. It's not clear in a lot of people's mind. Everyone thinks, well, we just want to go back to the state it was before. It was when it was pristine, when it wasn't disturbed by humans. And that's a fallacy. There are no pristine states. There was never a static state where species distribution stayed still and the environment didn't change. So any selection of a historic target state for restoration is arbitrary. You can do whatever you like. But there are some implications when we think about the fact that environments change. In a chapter with Patrick Osborne from the UK, we explored those. We said, what are the implications for reintroductions of this kind of environmental change? We came up with 4. So where things were in the past, their historical locations, doesn't mean that that is still suitable habitat today. Where things are now doesn't mean that that's the best place they could be. They could be restricted there for other reasons. Where they're missing, so the flip side, where they're not doesn't mean it's not suitable. There may be reasons why they can't access that area. And where they are now doesn't mean that it's always going to be good for them. We can illustrate that again with some examples, some New Zealand examples. So New Zealand and exotic predators. So this is just the increasing distribution of possums in New Zealand. The arrival of predators has changed habitat suitability. It's changed that environment for species. So if we look at distribution maps like this for kiwi and for mohua, yellowhead, the pale blue tells you where they were pre-human. So this is from fossil sites. The dark blue is where that distribution had shrunk to by about the 1970s. And the black is a more recent one. And we may have to redraw the map for mohua if we're not careful in the future. So just because they were distributed over that large area doesn't mean that those areas are now still suitable for them. They're filled with predators. That habitat suitability's changed. The other extinction hotspots, Hawaii. We look at the distribution in pale here of Hawaiian endemics up in these highland areas. You could say, well, that's their preferred habitat. They like to be up high. But in fact, these birds were once distributed right throughout lowland areas. That was their preferred habitat. But what's happened is introduction of a mosquito carrying avian malaria has changed that habitat suitability. And these birds are now restricted to these little upper islands where it's relatively too cool for the mosquito. And of course, the big one is climate change. Climate change changes everything, especially the rate of change. So we're looking here at a very long-term record, millions of years in the past. We can zero in just on more recent. So these are not predictions. This is data about changes in CO2 and changes in temperature. So this is fundamentally changing the habitat suitability for species. And we need to figure out how to deal with that. I explored the implications of some of these in a paper back in 2010. And I made the point that I felt we're moving away from the dictates of historical species distribution records towards inclusion of more risky interventions. That is, moving things outside of where they used to be. And I knew this was going to be a bit controversial. And you know you're in trouble when your name appears in the title of a paper. But nevertheless, some of these ideas were taken up when the World Conservation Union revised its guidelines. So importantly here, we've got guidelines for reintroductions and other conservation translocations. And those other conservation translocations are what we would call conservation introductions. So that is moving species outside of their historical distribution range for conservation purposes. We can think about a couple of those there, assisted colonisation and ecological replacement. So with some New Zealand examples, we think about what assisted colonisation is. That's where habitat suitability changes so much or so fast that species are unable to stay and adapt, or unable, perhaps because of barriers, to move into more suitable habitat elsewhere. So you must assist their colonisation by moving them from one place to another outside of their distribution. So example is the stitchbird or hihi. Modelling has shown that those remnant populations, or only 6 populations left, 5 of them exist from translocations, are at high risk of extinction unless we start to move them away from their traditional or their historical range. And with tuatara, which has temperature-dependent sex determinations, very sensitive to ambient temperatures, a case has been made for assisted colonisation as those temperatures change. Been talking about species, in isolation, protecting, conserving, restoring individual species, but we can think about the role that some animals play within their environment. So this flowchart is really just talking about the effect that wolves had or have within an ecosystem such as Yellowstone. So wolves were eradicated from Yellowstone National Park. As a result of that, the elk that they preyed on, their populations boomed, there was overgrazing, and there was severe erosion. The reintroduction of wolves restored that balance, that natural predator-prey process, and restored that ecosystem to some extent. That's all very well. We had wolves, we could put them back. What happens, though, when you've lost something through extinction? You've lost one of these ecological engineers. You can't just put it back. We're talking about ecological replacements. That is, finding some kind of substitute that you could put back in there to do that same job. And the most work around ecological replacements has been in replacing mega herbivores, these giant tortoises on island systems. One of these is, you see, Lonesome George, so another last individual that got lucky enough to get a name, appeared in David Attenborough's Galapagos program. So the loss of tortoises from these profoundly changes that ecosystem because of the way tortoises graze and trample that vegetation. With a colleague, Christine Griffiths, who's done a lot of work in this area, we tried to summarise this for Round Island in Mauritius. So we started off in a historical setting with tortoises. The tortoises became extinct, and a series of introductions of exotic grazers and exotic vegetation completely degraded and changed that environment. The restoration process involved taking those goats and those rabbits off, so these red areas here, taking them off, and restoring some of the natural vegetation, the vegetation that should have been there. But the trouble is, you had ongoing kind of weeding problems. The natural vegetation couldn't compete with the weedy species. So by bringing in an ecological replacement, a giant tortoise that wasn't originally there, to graze that vegetation, they've actually been able to restore that habitat to some semblance of its original form. We can summarize all this in thinking about a conservation translocation spectrum. So we start off with, at the top, that black box, and we ask the question, is the release taking place within the indigenous range or within that historical distribution? If it's yes, then what we're doing is one of two things. We could be adding individuals to an existing population, such as black stilt, that would be a reinforcement, or adding them into an area where they used to be but are now missing, such as frogs or the wolf example. If you're not putting it back into their indigenous range, you're doing a conservation introduction. You might be doing an assisted colonisation. So the example here is the Tasmanian devil. Populations dropping because of devil facial tumour disease, so that they've used assisted colonisation to create a disease-free population on an island where they never existed. And then these ecological replacements. Ideally, you want to keep the bits there. And extinctions are a loss. You can try and find replacements. Maybe replacements don't exist. It would be much better to have that original form back. And there are some people that suggest that we might be able to do that. So this sort of hit New Zealand scene because of Trevor Mallard, who's currently fighting for his political career. I think like 378 votes stand between him and oblivion. And he was asked to kind of future think for his electorate, Wainuiomata, and he he thought one of the things might be nice to have around for various reasons would be moa, and to de-extinct that moa. And the reaction was, you know, you must be crazy. Where did this come from? You've been reading too much Michael Crichton or something like that. But in fact, what Trevor had been reading was National Geographic. National Geographic has kind of teamed up with a group called the Long Now Foundation, headed by Stuart Brand. And Stuart has said, humans have made a huge hole in nature in the last 10,000 years. We have the ability now, and maybe the moral obligation, to repair some of that damage, to resurrect species. So we have the ability now. Do we have the ability now? Well, there are 3 pathways to de-extinction. The first one is a bit of a cheat. What you do is you find a near living relative. So in this case, people are trying to resurrect the auroch, this giant black beast that used to roam through the forests and the verges in Europe. I think the last auroch officially died in Poland in about 1627. So what they're finding is existing domestic cattle species, such as heck cattle, and you selectively breed them for these primitive traits. You make them bigger and bigger horns and darker, and what you end up with is a pretty impressive, if fairly docile looking beast. But is it an auroch? Have you really de-extincted something? Probably not. You get much closer if you think about cloning technology. So this is a bucado. It's the last bucado, so it has a name, Celia. Celia died in 2000. Unfortunately, in 1999, they took a sample of cells from her ear and they cryopreserved those. And it allowed people to attempt to clone her. So the procedure was to take those cells, or take a cell, take the nucleus out of that cell, and put it into a host cell that had been enucleated from just a goat, a surrogate goat. You do something to that cell, you zap it or whatever, and you make it think it's an embryo. It starts to divide, and you implant it into a surrogate. And then that surrogate will give birth to a live bucado. And they actually achieved this. So we could say de-extinction has already happened. Unfortunately, it only happened for 7 minutes. There was a gross deformity. So that clone died very, very rapidly. But a lot of the technical challenges had been met in doing that. The third way is where you can go back further in history, before people were cryopreserving anything. So you have ancient DNA. And ancient DNA is an unstable molecule. It breaks down very quickly. So if you go any distance back, it's very hard to read it. It's like people have likened it to reading a book, a very old book that's falling apart, so pages are missing. Sometimes you get a word or a sentence or a fragment. Thanks to a bit of desktop wizardry from George Church, who's a Harvard geneticist, it's possible to fill in those gaps, if you like. So what people have been doing, and here's Martha again, passenger pigeon, is read as much of the passenger pigeon genome as you can. So I think they've sequenced about 40 birds. There are 1,500 specimens out there, but there are gaps. You then compare that to a near relative, in this case the band-tailed pigeon, and then through this machine of George Church's, you fill in those gaps and you recreate something. There are a few other steps, of course, to ending up with a hatchling that's a passenger pigeon. Passenger pigeon's an interesting one to put all this effort into. They were a hyper-abundant species. 5 billion birds used to fly around. No one thought they could go extinct. There were stories of flocks that were 430 kilometers long, that took 3 days to pass, that might have had 3 billion birds in them. The record, someone fired into a flock with one shot and hit 15 birds. They were shooting them so much. They were using feathers, they were using the meat, but so many were dying, they were actually using them to fill potholes in roads. So no one really believed that they could make this thing extinct. And I'm not sure we want to de-extinct it either. I mean, we get upset when one pigeon sits above our car. Are we really ready for 1 billion? And it did make me think, what are the de-extinction candidates? And I went on to the Long Now Foundation, and they have this list of various things there. So passenger pigeon at the top, and not surprisingly, moa is in there, dodo, the auroch, bucado, down through woolly mammoths, saber-toothed cats, things like that. You recognize something in this list, we've got taxonomic bias, so we're really thinking about these big charismatic species. So how do you pick? What's a better candidate than another? It's something like the thylacine, the Tasmanian tiger. Is it really a good de-extinction candidate? It occurred to me that if you don't want to do this in order to create curiosities in a lab or a zoo, then you need to be releasing these back out into the wild. And that brings it into my territory, because what you're doing is you're reintroducing it. And we have some nice guidelines to think about that. So with colleagues, Axel in Canada and John in the UK, we took those IUCN guidelines, we turned them into a 10-point checklist that would be a pre-selection process for any de-extinction candidate. Before you even go to the expense or the trouble of trying to resurrect something, run it through this list. Do you understand why it went extinct so it doesn't go extinct again? What are the new threats that might be out there? Is there even habitat for it? Can you sustain that habitat long into the future? What do people think about it? Are people going to be on side with you with this or not? Is it going to affect livelihoods? And we ran a number of species through that. So thylacine actually came out as a possibility. Trevor Mallard talked about moa, and people said, well, the moa's not a goer, but he has at least one supporter. So this is Mike Dickerson, who's the curator in Whanganui Museum, and he oversees the largest collection of moa bones and parts. And Mike thinks that moa could be a viable de-extinction candidate. He said, I think it could happen in about 100 years, which nicely takes it outside of his lifetime, so we can't prove him wrong there. A lot of arguments, pros and cons around this. It's a controversial topic. So this is just a very brief snapshot. So down the left-hand side there, these kind of quest for redemption, moral imperatives, righting past wrongs. You could ask, how far back in history are we still responsible for? And on the other side are more compelling ones, I think. This idea of deep ecological enrichment. So not in the lab or zoo, but out there in the wild. Enhance the resilience of ecosystems. Restore those kinds of functions. There is a flip side, though. There are a lot of cons to it. And this poster actually captures at least 3 of them quite nicely. So people consider this to be a distraction from real issues. It's not as if we've solved all these other problems. It's not as if we don't have species on the verge of extinction. Much better to stop them going extinct than to try and bring things back. There's only a limited funding out there. Is this a distraction? Also in terms of funding and resources, too much money going into these things, you could argue. And I think an important one is this changing the public perception that extinction may not be forever. We're not in a crisis mode. We bring these things back later when things have improved. I think also on this we can think about ethical and moral issues. So this little cartoon here, this idea of close enough, this is actually a quote from Stuart Brand. People have asked him, saying, you know, how close could you get back to the original? And he said, well, close enough. But we've seen with Celia's clone, only lived for 7 minutes because it couldn't breathe. So there are kind of moral, ethical, animal rights issues around that. At the end of the day, though, this is a really cool idea. I think everyone can agree with that. There's a prominent Australian ecologist who's done a lot about prioritising species actions and argues against de-extinction because of the waste of resources. But he wrote to me in an email recently and he said, I would pay $1,000 to see a resurrected moa or woolly mammoth. So I don't think we can ignore that. I want to come back to this idea of our sort of humans' alienation from nature. This is Daniel Pauly, who's a well-known fisheries biologist, and he coined the term shifting baselines. He was talking about fisheries, but it's now being applied much more widely. His argument was that each generation, whether fishery managers or just us, gets used to a more impoverished environment around us. It resets our expectations lower and lower and lower. So we have this kind of shifting baseline of what the world around us should look like. With apologies to Daniel for the title for the next slide, but I call this Shifting Waistlines. So this is the Royal Guard in Saudi Arabia overseeing release of oryx. And as Keith explained, Yolanda and I spent a number of years in Saudi Arabia. And one of the things that was really interesting to work there as a wildlife biologist, conservation biologist, was a society that's gone through such profound social, economic change over such a short time. And some of those changes obviously been, you know, access to better weapons and better nutrition, but it's also around the idea that people's engagement with the world around them has changed. So we think only back in the 1950s, most of that population, the majority of that population, were nomadic herdsmen. Out there, engaged, their day-to-day livelihood depended on them understanding that natural world around them. And there's been increasing urbanisation, so more and more people are living in cities. With Saudi colleagues, we were interested to see how this had changed people's perception of their natural heritage, really. So we posed a questionnaire to a number of urban-based high school children. And we gave them names of species like baboon or gazelle or whatever, and we asked them to pick a word to best describe it, whether it's just kind of beautiful or amusing or tasty or something like that. And we're surprised one of the responses, around 50% of people considered the desert fox to be dangerous. This terrible, dangerous animal that's out there. It makes you think, what on earth is this desert fox? Is it like a wolverine? It's kind of all snarling teeth? Well, at great personal risk, I managed to get a photo of a desert fox. And here's the savage beast. And you can see she's holding a fox. Seriously though, we did engage with Bedouin rangers, people in the desert. They're not afraid of desert foxes. They understand how foxes fit into that environment. So this is kind of an alienation from nature and a lack of understanding. Not meaning to pick on Saudi Arabia, I'm sure we could do that same kind of exercise with very urbanized populations anywhere in the world, including New Zealand. Yolanda and I wrote a chapter for a book, came out last year, entitled Reversing the Extinction of Experience. So this is a term to do with engagement with nature and how we're missing out on these opportunities to engage with the natural world. And we argued that actual conservation translocations, reintroductions are a way of restoring a balance, re-engaging people with their natural heritage in a way to make them understand what it is that they nearly lost. And we see a lot of this in New Zealand, a lot of New Zealand action to feel quite encouraged and proud about. So just on our doorstep here is Orokonui Sanctuary, where it provides a place where natural vegetation is being restored, natural indigenous species are being reintroduced, and it's a place where people can go, they can see what the world around them should look like, and reassess their expectations for what the world should be like. I just want to wrap up now with some kind of thanks, acknowledgements, and some reflections, if you like. So just thinking about kind of my journey at Otago and how it's been almost surreal to have gone from kind of student to academic. This is Lloyd Davis, who was my PhD supervisor, and it's been fantastic to have been his student and then become his colleague. And it made me think about another kind of learning situation that I've been in through my life. So ever since I was an undergraduate, I've been studying karate, martial arts. And I think there are a lot of kind of parallels between that kind of teacher-student relationship and what we do within the martial arts. So in the same way that a PhD supervisor instilled those hard lessons onto their pupil, it's the same within karate, where teachers are instilling those hard lessons on their pupils. And then that student goes ahead, learns, gets students of their own, and passes that information on as well. I want to get back in Yolanda's good graces by thanking her for being my team. Probably not the best photo to show, actually. She's standing there wondering why I'm taking a photo of her rather than pulling her out of the mud of the Tasman Delta. But we've had some adventures together. We've always been a strong team, and she stuck by me. People change in marriage, don't they? They change. You go through phases. I know I've gone through some phases in marriage. But, you know, you get through that, you get over that. And now my kind of rock and my foundation is my family. So with my boys, Connor and Jasper. I want also to acknowledge the original Professor Seddon, who's with me here today, and my mother, who passed away a few years ago. It was really my father's tales of student life in Dunedin when he was here as a medical student that made me say, right, that's where I'm going to go. And I've been associated with the university for a number of years now. So this is the honours class from 1984. And there might be a few familiar faces in there. People have gone on to good things. Actually, this is outside the front of the Zoology building, and my first encounter with zoology was actually in 1981, during the orientation week. And I looked at this lovely facade there, and I thought I could probably climb that. So I did. I climbed that, and very soon the head of department's head appeared out of the window I'd just climbed past and ordered me down. I thought this is a bad start to university, and I want his career in zoology. But it was with great pleasure that in 2001 we could come back and rejoin the Department of Zoology, which has to be the most, or certainly one of the most collegial departments there. So it is my daily pleasure to interact with these smart, enthusiastic, friendly colleagues. And I do want to single one person out. So this is Ian Jameson looking very grim, saving another species that's about to become extinct. Ian's been my friend and colleague for decades now. And when I came to Otago, I actually approached him. I said, would you be my mentor for that? And he took that role on. So thank you for that, Ian, and you continue to inspire. Harleen talked about students. This is our energy, our enthusiasm, our productivity. So this is a selection of postgraduate students I've had the fortune to interact with, work with. So if you're looking at this and your photo's not there and you think it should be, then you need to change your Facebook privacy settings. And when I came here, it was to take up the directorship of the wildlife management program. And it's each year a new cohort of smart, enthusiastic, passionate people I have the pleasure to interact with. And it's going to be these people that are going to go out and become the wildlife managers, the researchers, the conservation biologists of the future. And these are the ones that are going to help us kind of reconnect with our natural heritage. And I can only say I think we're in very good hands. Thank you very much.
Ladies and gentlemen, good evening. Tēnā koutou katoa. My name is Alison Cree, and it's my great pleasure as the Head of Department of Zoology to thank Phil on your behalf for his wonderful talk tonight. You will have learned much about the history of the science of reintroduction biology from tonight's talk. From the time when, staggeringly, it was possible to fill holes in the road with passenger pigeons, something I'd never known before, to recognition that extinction was a reality and was going to be a continuing phenomenon if action was not taken. We've learnt a lot tonight about the history of translocation as one of the actions that can be taken to reduce that risk of extinction. And I hope that you will have appreciated in the history that Phil's given tonight what a leading role he is playing in today's use of translocations through the IUCN specialist groups, of which he's a member of quite a few, to take us forward into issues like ecological replacement and the de-extinction debate that's going to be ongoing, no doubt, in future years. So it's been fascinating to learn about that history from your research, and not just research in terms of thinking about the issues, but putting them into action, and we applaud that work, Phil. I would also like to note the significant role that Phil plays in the university community and the service he's contributed to the University of Otago and to his role in our department. Phil is highly regarded for his leadership of the wildlife management program. You've heard about how he's been the director of that program since he arrived in our department in 2001. And he continues to refresh and keep that internationally attractive program alive with wonderful students. So thank you, Phil, and your team for the work in that area. You've heard also about Phil's role as a supervisor of postgraduate students, and he has many of them, and they're really productive. And we appreciate that greatly. Finally, I would just like to acknowledge the very calm and lucid and collected way in which Phil, as a member of our department, helps us analyse issues. That's a really important skill, and we thank you very, very much for that, Phil. It's my great pleasure now to hand over, as a token of appreciation, a gift from the university, and thank you once again, and invite everyone present to join us to celebrate with Phil in person in the staff club at the end of the function tonight. Thank you.